Polysaccharide is in carbohydrates. Uh, we can say on hydrolysis, it will give you an more than 10 monomeric units. On hydrolysis, it can give you more than 10 monomeric units. Okay, fine. So what are the polysaccharides you have? One is starch, glycogen. Okay, one is starch, second one is glycogen, third one is cellulose. We need to complete carbohydrates today, so please copy down fast. Okay, we have three types of polysaccharides as per our syllabus: starch, glycogen, and cellulose. What do you mean by starch? Starch. What do you mean by starch? You need to explain, like, it's a type of carbohydrates, in that it's a type of polysaccharide. Okay, it's a type of polysaccharide. It is present as plant storage food. Okay, plant storage food. Your starch is said as plant storage. Yes, Sujan, you are right. Okay, so it is called as plant storage food during during a non-photosynthetic time. During non-photosynthetic time, during non-photosynthetic time, it will give you an energy. During a non-photosynthetic time, your starch get hydrolyzed to give a energy. So then, when starch hydrolysis, if it is give monosaccharides, then what are the composition of monosaccharides present in your starch? That is the first and foremost question. What is the composition of starch of monomeric units present in your starch? It's an glucose. That too, it's an alpha glucose. Okay, can I able to get the point? It is an composition of alpha glucose monomeric units. Okay, fine. Next, it is insoluble in cold water. Cold water means to say it's just not a refrigerated water. One is hot water, one more is normal water. So it is not soluble in normal water also. It is white amorphous structure. Okay, it's a white amorphous structure. Yes, fine. It is not soluble in water and it's not soluble in alcohol too. Okay, it's not soluble in cold water and it's not soluble in alcohol too. It's an colorless, odorless solid. You can say colorless, odorless, crystalline solid. It is an colorless, odorless, crystalline solid. These are all the features of your starch. Okay, these are all the features of your starch. In the starch, okay, in starch structure, in structure of starch, we have two types. In structure of starch, we have two types. One is amylose. Second one is amylopectin. This is very important. You should show your full concentration. You should be attentive in the class. Please don't leave your mind here and there. Please focus on the class. Based on the structure of starch, you have two types. One is amylose and amylopectin. If you didn't understand, please inform me. Like, ma'am, can you repeat? Or, ma'am, I didn't understand. So, it could have been very useful for us to, or help me to understand you. Okay, so we have two types, that is amylose and amylopectin. So, what do you mean by amylose? So, I'll write here amylose. Amylose is an water soluble substances. Okay. In starch, these amylose are present in 15 to 20%. This amylose, in entire composition of amylose, it is present in 15 to 20%. Then it is an straight chain structure. Okay, it is an straight chain structure where it can link to 200 to 1000 glucose unit. It can join to 200 to 1000 glucose unit. That much length it have. That much length it have. Then it is linked by the glucose units are linked by glycosidic linkage. Yes, of course, right. C1 and C4. That glycosidic linkage is exist between C1 and C4. Are you getting my point? Yes, Nikhil, I'm also facing so much of network issue. That's why it's showing blur. From yesterday, it's a problem. I don't know why, what happened. So, this is an amylose. Amylose is water soluble, and in starch, in starch, 15 to 20% is covered by your amylose, and it is having a straight chain structure. That straight chain linked by 20 to 1000, sorry, 200 to 1000 glucose unit. It is a long chain, and that's linked. The glucose units are linked with an 1,4 C1 and C4 glycosidic linkage. Okay, are you getting my point? Can you able to understand? What Srujan, Gokul, Meghana? Can you able to understand? Because this is very important, very, very important. This will be asking for 3 marks or 4 marks question. But it starts explain in detail or explain in brief. They are going to ask this question. Okay. Next. Then I'll write this structure. I'll just write like this. This is some glucose units. Okay. These are the glucose units. It further linked like this. So this is your amylose structure. Amylose structure. You can fill this, no problem. So we are taking here alpha amylose. So you should remember that we are taking here an alpha amylose. Okay. So this is your amylose structure. Write it down. This is your amylose structure. Not clear, Kavya. That's what I'm saying. It's a network issues. Does it clear now? Does it clear now? It is just an alpha glucose units linking an alpha glucose by 1,4 glycosidic linkage. And just linking. Okay, Kavya, it's better, right? We just write the structure of alpha glucose and copy it down. This is amylose structure. Your starch composed of two types. One is amylose and amylopectin. This is structure of amylose. Shall I move further? Can okay, Megan. And sometimes it will show in the blur, but again it will take up the time. But if network is clear, then you can see it properly. Because of cloud and all things, it's not going good. Shall I move? Finish writing this? Okay. So let us write the second structure, that is amylopectin. Okay, fine. Amylopectin. Okay, Kavya is asking something. I'm sorry for the disturbing in between the class. No problem. Can you please send the running notes of yours in WhatsApp? These sheets. Which sheets you want? I can send, no problem. And I'll send slides too. No problem, Kavya, I'll send. But can you able to understand? Because I know my running notes are fantastic that I only can understand. If you can understand, it's no problem, I can send. So now, this is amylopectin, but you should write. This is the first thing you should write. If you are seeing my notes and if you are not writing yours, means you should write, right? Okay. During my class only, you should write. Then I can send it. No problem. So amylopectin. Okay. Amylopectin. Just a second, it's showing here. Yes. Perfect. So amylopectin, right? So amylopectin, it's an water insoluble. Water insoluble. class. They're saying they can't join for the class because of internet, I think so. What to do? Let us see. Let us continue class. So the previous one, what we read, amylose is an water soluble. But here, the amylopectin is an water insoluble. Okay, it's an water insoluble. And it is present 80 to 85% starch. Okay, 80 to 85% of your starch is combined, or its composition of your amylopectin. 15 to 20% is your amylose. 80 to 85% amylopectin, and it's an branched chain. Okay, and it's an branched chain, it's not an straight chain. So what we read in the amylose, it's a straight chain, it can compose of 200 to 1000 alpha glucose unit, and that is linked with 1,4 linkage. That is for amylose. Yes, right. But here it's totally ulta of amylose. That is, the amylose are soluble in water, and amylopectin is insoluble in water, and it's present 15 to 20%. It's present in 80 to 85%, and that is a straight chain. And this one is a branched chain, and that is linked with 1,4 carbon. This is linked with 1,6 carbon. Okay, this is linked with 1,6 glycosidic linkage. Are you getting my point? So, and the branched chain over there, it's composition with 200 to 1000, but here it's of 24 to 30 alpha glucose. In your branched chain, it can carry 24 to 30. You can write 25 to 30 units of glucose, alpha glucose are present in your branched chain. These are all the major two importance of your amylose and amylopectin, and they could have asked this for 3 marks question. Okay, this is an important question, and this will you ask for your 3 marks question. Fine, let us write the structure of it. So first we need to write amylose structure only, because if we want to branch it, we need a straight chain first. So I'm going to write this straight chain. This is your amylose, right? You need to write, you need to write, you need to write. And you fill this. In your examination, you should fill this. Otherwise, you didn't get the marks. Okay. So what I informed, it's a linkage of 1,6 glycosidic, right? 1,6 glycosidic linkage. So what I'll do, I'll just take it up and I link with, sorry, I'm sorry for moving it, like this. Again, here, it will move further. So this point is said it as glycosidic C1 and C6 linkage. Are you getting my point? So this is your amylose chain. This is straight chain which contain 200 to 1000 glucose units, that is to alpha glucose unit. When it starts branching up for the 1 and 6 carbon glycosidic linkage, it's convert the amylose to the linkage of your amylopectin, and this branched chain can convert 24 to 30 alpha glucose unit. Okay, can you able to understand? If it is only straight chain you have, if it is only linked straight chain you have, that is amylose. In that, if it is branching, that is amylopectin. So you should know one thing: in your starch, both are present. Some amylose are present, and that amylose leads to the formation of amylopectin. That's why the starch, whatever we are reading, the starch, a plant storage material that can be composition of amylose and amylopectin in the percent of 15 to 20 amylose and 80 to amylopectin. So in your starch, you have two types of linkage, glycosidic linkage. One is 1,4-glycosidic linkage. Second one is 1,6-glycosidic linkage. This what I told, 1,4 and 1,6. These are very important. This will be asking for your 2 marks. Okay, this question will ask you for 2 marks. So please note it down. Please write it down. Amylose is 1,4 linkage. Amylopectin means 1,6 linkage. The branching chain of your amylose is said it as amylopectin. Is said it as amylopectin. It's linked with 1,6-glycosidic linkage. We have only 10 minutes. So next, glycogen. So glycogen is as same like your starch, but it is animal storage. Okay, it's an animal storage. And you have more amylopectins when compared with your starch. When compared with your starch, you have more amylopectins. Okay. It can be called as animal starch. Animal starch. It's an animal for storage food and it can be called as animal starch. Okay. Are you getting my point? Just give me a second. I'll take up the attendance because 2 days back I forgot to take the attendance only. Please try to write your structures of amylose and amylopectin. Okay. So, okay, fine. So, we can write like this also. Usually, during our explanation, we will write like this. This is your, you need to write like this only, but it have more branching chain. So, in fastly, I'll explain the different starch and glycogen. Please note it down. Starch is your plant storage food. Glycogen, it's your animal storage food. Your starch having an amylose and amylopectin in the composition of 15 to 20 and 80 to 85. Have more amylopectin. Here in 20 to 30 units, you can see the branching. In 20 to 30 units of amylose, you can see the branching. Okay, but in glycogen, in 2 to 3 glucose units, you can see the branching. So the branching difference or the branching is more in your glycogen, is less in your starch. Okay, more in your glycogen, less in your starch. Fine. Next point. It is soluble in water. Sorry, your starch is insoluble in water. Glycogen is also insoluble in water. It is a composition of alpha glucose units. It's alpha glucose units. No problem. So this is the difference between your starch and glycogen. The major difference is how it's going to branch. The branches is amylopectin number is more in your glycogen and less in your starch. And for every 20 units, it means to say, for example, this is your glucose 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, like that. So for the 20th unit, you are going to see the branches where in your starch. But in your glycogen, what happened? For every 2 to 3 unit, you can see the branching. So the branching bunch is more in your glycogen when you are compared with starch. Okay, understood? Understood. So your glycogen and starch both are in the alpha glycosidic linkage. Okay, both are in alpha glycosidic linkage. So let us see your cellulose. Cellulose. So cellulose is also an polysaccharides. On its surface, it will give you more than 10 monomeric units, that is glucose unit. So in that you don't have any change. But it is present in the plant, that is for primary cell wall. It is present as primary cell wall. Okay, it is present as primary cell wall in your green plants, or you can say it's an waxing coating outer your plant cell. It is wax coating of your outer ring of your plant cell. About 33% of your plant cell, it's covered with cellulose. About 33% of your plant cell composed of cellulose. Okay. It's also linked with 1,4 C1 and C4 glycosidic. This is also composition of C1 and C4 glycosidic linkage, but it's an beta linkage. But it is an beta linkage. Why? Because we are using beta glucose. Okay, here beta glucose has been used. That's why the bond is beta bond. There is beta glycosidic linkage. So if you are writing the structure of glucose, I'll just show you in a fast way. Just please write it a neat structure, not like me. Okay, this is your cellulose. It doesn't having much more branches like amylose or amylopectin, whatever we are reading in the starch and glycogen. It having the linear structure of it, that is to a beta glucose or linked, that it leads to the formation of beta glycosidic bond. This is called it as cellulose. Cellulose is the composition of beta glucose. On hydrolysis, it will give beta glucose. On hydrolysis of your starch and glycogen, it will give alpha glucose. Hydrolysis means they'll just break up this bond. I'll explain that. Okay, I'll explain that. No problem. Hydrolysis, you don't have that actually, but on hydrolysis means if you are giving a water molecule with the presence of hydrogen, this bond going to be break up and this molecule will separate and it will give the glucose units. So in your starch and glycogen, it will give alpha glucose unit and your cellulose will give your beta glucose units, this much only. So this is about your polysaccharide. So let us see what are the biological importance. Biological importance of your carbohydrates. So what are the biological importance of carbohydrates? So yes, it will give you an energy. It will give you an energy because your starch or carbohydrates will give you an more energy when compared with the other biomolecules. It will give you an more energy and fast energy. Okay. So then in plants, it's help for the storage food. Storage food. In animal also it will help. So it's act as a storage food. Okay, it will act as a storage food. It will give the direct energy for your muscles. For your muscles, it will give direct energy for your muscles because in muscles it will store you as an glycogen. During our warm-ups, during our exercise, we want energy directly to the muscles. So that energy is directly supplied by the glycogen of stored in your muscles. So that's why it called it as direct energy source. Okay, direct energy source. And it will help the movement of the blood. Movement of blood. Because blood cell need to take up the oxygen, so that's why it should bind to the uh, what your hemoglobin. So if it is need to bind to your hemoglobin, it requires some energy. That energy has given by your carbohydrates compounds. So these are all the main biological importance of your carbohydrates. So, and it is present in your DNA like ribose. So it's your present in your RNA, and we are using it for drug resources and all. So I'll explain it tomorrow's class for about 5 minutes, and we'll take up with the protein. Okay. Then this session will end. Until and unless if you have any doubt, you can ask me. Biological importance and the sources of carbohydrates I'll explain again.